Hello, everyone. Today I'm going to talk about my work, MethPhaser, methylation-based haplotype phasing of human genomes. So long-read data have provided new insights for more comprehensive structural variations. You can see with short read, you can only count 10 to 12K SVs per human, but with long-read data, you can come about 2 times more than that. And it helps us access the repetitive regions, medical genes. Also, it can give us a better phasing or assembly data, like phasing N50s or less gaps. And on the right, you can see several papers showing that it helps accurate detection of the complex structural variations, and it helps assemble the complete sequence of human genome and phase the cancer-related samples. So I always come back to this great example that shows exactly why we need phasing. There's an example of a gene called serpin methyltransferase genes. It's on the it encodes the enzyme that metabolize serpin drugs. There are 2 variants on this gene, more than 8,000 base pairs apart. And when these 2 variants are on the cis relationship, which means these 2 variants are on the same copy or same haplotype on this gene, so the gene will produce intermediate metabolizer, which means the drug's movement will be affected but not disabled. But when these 2 variants are on the trans relationship, which means this gene will produce poor metabolizer, which means the drug's movement will be disabled. So when a doctor is trying to give a patient a medicine without knowing the phasing information between these 2 variants, they might give the patient drugs when these 2 variants are in the trans relationship, which will cause severe side effects. So after stating the importance of the phasing, there are 3 methods to phase the haplotypes. The first one is called population-based phasing. It phases human genome based on common variants. It requires a lot of cost and collection effort because it requires a large population's sequencing materials. And the second method is called trail-based phasing. It phases individuals' variants based on this individual's parents' variants. So it can phase common variants, and it can phase rare variants that also being carried on this individual's parents. But it also requires lots of cost and collection effort because it needs this individual's parents' sequencing materials. And the method I like to emphasize here is called long-read-based phasing. It uses this statistical method to phase these individual's variants. So it can phase common variants, rare variants, and normal variants that only exist on this individual. But the issue with that is using this statistical method cannot fully phase this individual's variants. So that means long-read phasing does not solve everything. So you can see that with the read length increase, the phasing N50s are gaining, but the long-read-based phasing fails when the homozygosity hinders phasing. So in here, I'm showing an example that exactly shows why when a long-read-based phasing fails. So on the bottom, you can see there are 2 ancillary phase blocks. Each ancillary phase block have several heterozygous variants. That helps the long-read-based phasing. But in the middle, there is a homozygous variants that are same on 2 haplotypes. And this unphased gaps is longer than the read length. So basically, the gaps remains unphased. The reads are being unphased. And the variant phasing information between those 2 ancillary phase blocks are remaining unknown. So basically, long read helps, but it does not solve it all. So methylation is another signal for read phasing. So allele-specific methylation exists on human genome. And here, you can see that the red dots means methylations on the human samples. They are different on different haplotypes, and MethPhaser is using this exactly allele-specific methylation signals as an addition to the traditional SNV-based signal to improve the SNV-based long-read phasing. So you can see that with MethPhaser, it actually improved the phase block length by a lot on H002 sample. The blue bars are the SNV-based phase block and 50. And you can see with MethPhaser, it actually increased the phase block and 50 by a lot with the H002 sample with different chemistries. So to dive more into MethPhaser's algorithm, it first calculates the haplotype-specific methylation CPG locations, and it based on that iteratively haplotype reads with methylation. And when we have enough reads in those unphased regions being haplotyped into the haplotypes, we can use those reads to bridge the unphased region based on those newly haplotyped reads. So on the right, we first have an isomate phasing result. You can see there are two isomate phase blocks. With non-polar reads, we have methylation signals directly reported along with the reads. And in the middle, there are unphased regions with unphased reads with methylation signal also reported. But with MethPhaser, we use message signal to assign those reads to different haplotypes, and we actually connect those two fragmented isomate phase blocks together to form a much larger one. So with that, I would like to show the improvement of MethPhaser on H302. So the input data for MethPhaser requires phase sensitivity and band file. It only works on autosomes because the random X inactivation. And you can see on the left with different chemistries, with different sequencing kits, MethPhaser increased the N50 number by a lot. With on the right, you can see with switch error and flip rate, which means the wrong assignment of single pair of the isomeres and the two consecutive wrong assignment of two isomeres. MethPhaser both gave us a very small phasing error increase. So after seeing we improved the N50 length of the phase blocks. So does MethPhaser actually improved on some meaningful regions? The answer is yes. So MethPhaser improved medically relevant regions. So on the left, there's a table showing that MethPhaser actually gave us more phase medically relevant genes with less phase block number needed, which means we not only phase more medically relevant genes, but we make those genes more consistent because we require less phase blocks to cover those genes. So on the right, I'm showing an example of HLA genes. So this is an example showing that how MethPhaser connects the HLE and HLC genes. So just assume we have a variant on HLE and another variant on HLC. So with isolated phase block, we cannot know the haplotype information of those two variants because there are two gaps in the middle and the information is fragmented. But with MethPhaser, we can actually connect those isolated phase block, and further, we can know the phasing information of the variants in HLE and HLC. So on the top, you can see in the IGV plot, there are two large pile of unphased reads in the middle, which gives us the unphased gaps. But with MethPhaser, with messenger signal, we actually can assign those reads to haplotypes. And on the right, you can also see a positional usage of MethPhaser is to identify the haplotype-specific structural variations. There was a haplotype-specific insertion in the unphased reads, and MethPhaser actually can accurately classify those into one of the haplotypes. So we test our robustness on other samples and populations. We test on MethPhaser on HPRC sample with ultra-long reads. We can reduce the phase block number from 900 to 600. And on the right, we have less optimal patient samples from blood. It improves the N50 still, but because the read lengths are smaller and the phase block N50 are smaller, so we can also reduce the phase block number by by more than 2,000. So to conclude, MethPhaser is the first method that combines non-read epigenomic and genomic variants for genome scale phasing. MethPhaser achieves 1.5 to 3x phase block N50 length boost against ICV-based methods on human samples. It will rescue previously unhaplotype reads, and MethPhaser can be directly attached to traditional ICV-based pipeline for great improvement with little cost. And I'd like to thank my advisor, Dr. Tao Trangen at Rice University, and Sergey Agnezov as a senior scientist at Dandapur when I was an intern at Dandapur, and Dr. Frista Legic who also supervised this project at the Baylor College of Medicine Human Genome Sequencing Center, and Mohammed, who performed the analysis on the blood sample. And the method is freely available online.